meaning, this is what is a cell. It is the basic structural and functional unit of living organisms. So if you are going to cut yourself into smaller pieces in a way that the smallest pieces would be functional, then we call that small pieces as cells. Okay? So cells make up living things and carry out activities that keep a living thing alive. So what makes you alive, you and me alive, is because of these fundamental units called cells. Now, there were people long before who combined themselves in proposing this cell theory. So what is this? Cell theory, this is a collection of ideas, conclusions from many different scientists over time that describe cells and how cells operate. So we have these three contributions of this scientists to propose this cell theory. So first, we have here, all non-living things are made up of one or more cells. So if you consider yourself as living, then whether you like it or not, you are composed of cells. So all living cells arise from pre-existing cells by division. Now accordingly, these cells come from pre-existing cells. Of course, when you talk about evolution, accordingly, we all come from a single cell, according to them. And this single cell keep dividing and dividing and increasing its complexity, and of course, even in the in the structure, enhancing its structure, say from simple, which is prokaryotic, becomes eukaryotic, which is already more complex than the other one. Now, but of course, when we go into creation, now, when was life start? Of course, that starts from creation. God created all things, and the last He created was man, human, Adam and Eve. And they already have consisted of cells. And of course, these cells, accordingly, come from pre-existing cells by division. But of course, not all cells in our body would undergo division. But for our skin cells, they keep on dividing. Okay. Now the cell is the basic unit of structure and function in all living organisms. So the smallest functional unit of our body, which do all the works, is called cell. So these are the contributors of the cell theory by Robert Hooke, when he first discovered a dead cell on the cork. That was in 1665. And of course, this was made possible because of the invention of Anton van Leeuwenhoek. Now, Leeuwenhoek observed living cell in his invention called the compound microscope. So he's the one who invented the compound microscope, and that was used by Robert Hooke. And we also, that was in 1674, and in 1883 by Robert Brown, he discovered later on nucleus. And this opens up to the discovery of DNA. But it takes several years for the DNA to discover, from the discovery of Robert Brown. Now there we have Now these are unicellular organisms, meaning one organism composed only of one cell. We have another, the multicellular organisms, meaning one organism is made up of more than one cell. Multi, you have the word multi. Like for example, plants, then the fungus or the fungi, and then the animals, so that includes human. We are all multicellular. So if you try to look at the cell in a microscope, then you have that multicellular organism, the plant. That one is the plant. So you can see here several cells. So you have here very distinct. Now the green one are the chloroplasts. So very distinct. And which comprising the green pigment called the chlorophyll. And you have also another example of a cell, the muscle cell. So this is your muscle. This is a muscle, so with striations. So you could see several, cells here. So that is for multicellular organism. And how about the size? The size of the cells vary from 1 micrometer to the largest egg of the ostrich to which is with an average size of 18 centimeter. That is still cell. So you have the body cells, our body cells, which measure around 1 micrometer, or say you also have the smallest cell like the bacterial cell, the one that causes tuberculosis, that's very, very small. So Mycobacterium tuberculosis, they are very, very small. That's why it can be carried by air for transmission. So it's airborne, very, very small. And we also have this. So you have here mga mycoplasma, or say mga 0.1 micrometer, like Mycobacterium tuberculosis. They are very, very small. And we have the largest cell, the egg of the ostrich. Your chicken egg is just smaller than the ostrich egg, and that is still cell. Then we have the sperm cell, which with an average size of 5 micrometer, and of course the egg cell or your ovum, our ovum, that is very, very large compared to the sperm cell. So that sperm is only 5 micrometer, whereas this egg cell is 120 micrometer. So that black ones there, those are millions of sperm cells that are wanting to get inside the egg or the ovum. And we have the longest cell. What makes it long? Because of its finger-like projections called the dendrites. That one. We have the nerve cell, or we term that as neuron. It has a size of around 1 meter. Okay? Because of its finger-like projections. Now, of course, we have the different shapes. It has this disc. So biconcave for the red blood cells, then the nerve cells branched, then white blood cells, they are bigger than the red blood cells. So they have different shape also. So they change shape when they engulf microorganisms. Okay? So they become large. These are the soldiers of our body. And of course, we have now the structure of our cell, which using the electron microscope. So the electron microscope will magnify your cell or the object to be viewed up to these times. Pila ni? 500,000 times. So that 0.1 micrometer that can be observed under the electron microscope, so it can be viewed or enlarged up to 500,000 times. So that's why it can be observed already. Now for the compound microscope, we have here, this one we don't have, but we only have here the compound microscope. It can only view the object up to 2,000 times. Okay. Now this is an example of an animal cell with all those structures. Now this one, we have the largest. Now generally, the cell is divided into two, or say maybe three. The outer part is the cell membrane. Then you have the inside, the cytoplasm, and then of course these are the cytoplasmic organelles which are embedded here, and we have the nucleus. That purple is the nucleus. But of course we have those details of the parts of the animal cell. Now you can see now the difference between the plant and the animal cells. So as you could see here, animal cells don't have the cell wall. Animal cells don't have this structure called the chloroplast. And the plant cells have bigger vacuoles. You could see here, majority of the area of what is inside of the cell is made up of this vacuole. Whereas these vacuoles here are just very small. So it has vacuole, but it's not that very distinct. Okay. So you have here the bacterial cell. So bacterial cell or prokaryotic cell, it has simple structure only and it lacks several organelles. But it contains the following: the capsule, this one, the capsule, and of course the cell wall. So made up of cell wall. So, and then it has the cell membrane, that one, yellow, and then you have the cytoplasm, and then you have the flagellum, food granule. It contains DNA plus. It contains DNA, and then you have the ribosomes and the nucleoid. Now nucleoid are not true nucleus, because bacterial cell lacks nuclear membrane or the nuclear envelope. That's why it doesn't have true nucleus. Now we term this region where we could find the DNA or the chromosome as the nucleoid. Nucleoid is not a true nucleus because it doesn't have the nuclear envelope. And you have here the pili. Pili plural, pilos singular. Then this one, the flagellum. This one, mga hair-like structures. Okay. So you have here, so as what I had said, generally the cell contains the following: the plasma membrane, the nucleus, and the cytoplasm. And you have here the cytoplasm consisting of the following. You have the liquid part of the cell, we call the part of the cytoplasm called the cytosol. That's the fluid part. The fluid part of the cytoplasm. And another, you have the different cell organelles. Okay, so let's talk about the plasma membrane. Now, plasma membrane, where you could find this? Now, see a cellophane-like structure called the cell membrane or the plasma membrane. And that's, when you look at it under the electron microscope, that's how it looks like. Oh. So it's a complex structure. Now, it's made up of semi-permeable, semi-permeable cell membrane is, or plasma membrane is semi-permeable. So it's made up of 2 layers of phospholipid bilayer. Okay. So it's said to be, when we say semi-permeable, it allows certain materials only that are needed by the cell. Now, other materials are not allowed to get inside. So it filters what are those molecules that need to be taken in. That is what called a semi-permeable, and that membrane is semi-permeable. And it's made up of 2 layers of phospholipid bilayer, lipid molecules with protein, phospholipid, in which protein molecules are floating. So this is one layer, and this is another layer. So the blue, the balloon-like structure is the head of the first layer of phospholipid, that is the phosphate group, and the red one is the lipid group, and this is another lipid group, which is the tail, and this one is the head or the phosphate group again. So you have here the 2 layers. This one is one layer, and this one is another layer of phospholipid. Okay. Now what is then the role of this phospholipid bilayer? Now, by the way, this phospholipid bilayer is amphipathic. Now, when you say amphipathic, this contains the hydrophobic region and the hydrophilic region. When we say hydrophobic, hydro-water, phobic, phobia, afraid of water. So this tail part, the red one, is said to be, because this is lipid, this is insoluble in water, so this is phobia of water. So this is the hydrophobic region. And the balloon-like, this is the phosphate, which is the hydrophilic water lover, the hydrophilic. So the whole thing here is amphipathic. Amphipathic, meaning made up of the hydrophilic region, afraid of water lover, and the tail part, which is the hydrophobic region. This is the lipid group. So this is the hydrophobic region. Now, what is the role then of this plasma membrane that maintains shape and size of the cell, protects internal contents of the cell, regulates entry and exit of substances in and out of the cell, and that maintains homeostasis. Now, generally, the role of this phospholipid bilayer is to facilitate the transport of molecules or small and uncharged particles. This one. All those small and uncharged particles, meaning without charge, will pass through here in the phospholipid bilayer. Like, for example, water. Water could pass through here in between this phospholipid bilayer. It can pass through here. And all those small and uncharged particles will pass through here in the phospholipid bilayer. Now, how about this? Large molecules. Now, these large molecules are proteins. Now, these proteins facilitate in the transport of large and charged particles. So, those which cannot pass through in this phospholipid bilayer, because they are large and charged, meaning with charge, like ions, sodium, calcium, potassium, they pass through here in the protein layer, or say in the protein molecules. Now, what this green, this is another protein. This is another protein which attaches carbohydrates. So this is the glycoprotein, another glycoprotein. Glyco means carbohydrates linked with the protein molecules. And we also have the glycolipids or carbohydrates linked with the lipid molecule. But it's not observed here well. But there are some carbohydrates which are linked with lipids. We call them glycolipids. Now, regardless of where they attach into, now these carbohydrates functions because they are projecting outwardly. They function for cell-to-cell recognition. Now, cells know their neighbors. How about you? Do you know your neighbors? Are you taking care of your neighbors? Sometimes our neighbors are sick and say, did you extend help to them? Or maybe your roommates. Now you should know your neighbor's class. Because someday you too will get sick and you will be blessed with your neighbors. So you should know them. Know your neighbors. Now our cells know their neighbors. If there are, say, something that will get inside and that is to be introduced inside and they are not part of their neighbors, then they have to what? Reject it. They have to quarrel. You don't belong to this place because they are not their neighbors. And that is what is problem in organ transplant. So there are some transplant which are not successful. It's because maybe their carbohydrate structure is not or very far in terms of structure, very far, not similar. Of course, each individual has its own carbohydrate structure. What makes us unique, especially in our fingerprints, what makes it unique is because of the carbohydrate structure in our fingerprints. And that makes it unique, even in identical twins. So no two individuals are the same. So you should not compare yourself with others. Each of us are given unique characteristics for a purpose, for God's glory. So if you are good in singing, then others are good in preaching. If others are good in preaching and singing, then maybe you have big ears. So you can be a good listener to others' problems. So each is said to be unique. Okay? Now another part is the cell wall. Now cell wall is present only in plants and bacteria. In prokaryotes, they have cell wall. Okay? So you have here, this is that cell wall is made up of that polysaccharide called cellulose that is present in the fibers. The cellulose, that one. The cellulose, which is the polysaccharide. And of course, we have the hemicellulose. Now, what makes cell wall very strong is because of this polysaccharide, cellulose and hemicellulose. And of course, there's also pectin. And that contributes to the rigidity of the cell wall. Now, how do you define this? So, non-living and outermost covering of a cell of the plants and the bacteria, and they can be tough, rigid, and sometimes flexible. So, made up of cellulose, hemicellulose, and pectin, may be thin or thick. So, there are some which are thin and some thick. So, function that provides definite shape, strength, and rigidity that prevents desiccation among cells. You say desiccation, dry up. Helps in controlling the cell expansion and that protects cell from external pathogen. So aside from cell membrane, plant cells and bacterial cells are being protected by the cell wall. But for bacterial cells, they don't have that. Their cell wall is made up mostly of pectin, not cellulose. For plant cells, they are made up mostly of cellulose and hemicellulose. Okay. Now another part is the nucleus. The nucleus is of course, this is the controller of the cell. Why? Because this is where you could find the DNA, which is the one controlling the cell activities. So it's located near the center of the cell with that diameter, and this is present in all the cells except for red blood cells and sheep tube cells. Red blood cells don't have the nucleus. And of course, the bacterial cells or prokaryotes don't have the nucleus. Now, this structure is well developed in plant and animals, and underdeveloped in bacteria and the blue-green algae or the cyanobacteria, or these are prokaryotic. These are prokaryotic cells. And most of the cells are uninucleated, meaning having only one nucleus. One cell, one nucleus. Now, few types of cells have more than one, like the skeletal muscles. Kanang inyong unod. No? Kanang unod, flesh. It's the skeletal muscle. Isa ka cell made up of several nuclei. Now, nucleus has a double-layered covering called the nuclear membrane. This is absent in prokaryotes. It has pores. So, meaning, since it has pores, so it allows molecules to get in or out of the nucleus onto the cytoplasm. So, we have, this is where you could find the DNA or the deoxyribonucleic acid. And of course, which is enclosed in a chromosome. So you have here the chromatin materials. These are the chromatin materials, and mga fiber-like. Now, when the cell divides, these chromatin materials appear as chromosomes. Mga mo na siyang chromosomes. So if you try to look at this under the microscope, these are made up of DNAs and proteins. They condense many chromatin during cell division. Now, when the cell does not divide, you cannot see chromosomes. You can see only chromatin. But when this chromatin condense, then that's the time you could see chromosomes. Okay. So now chromosomes contain the stretches of DNA called genes. Now, a portion or the length of the DNA that codes for a protein is called the gene. The gene is the hereditary unit. So that causes why your nose looks like the nose of your father or maybe your mother. Because the genes for the nose of your father was then carried to the sperm cell that fertilizes the egg of your mother that makes up you. So that is the gene, the hereditary unit. So function, control all the cell activities. Then ang nucleus synthesizes RNA or ribonucleic acid. And of course the function that stores hereditary information in the genes. So usually all the necessary genes are found in the nuclear chromosomes. So tanan na makes the major except for some traits no? Like maternal traits are found in the mitochondrial DNAs. But for the chromosomal DNAs or nuclear DNAs, I should say, no? The nuclear DNAs, they are found here.